much for including me in this forum. My name is Yassmine Akkari. I am the Senior Director in the Clinical Laboratory of the Institute for Genomic Medicine at Nationwide Children's and Professor of Pathology at The Ohio State University. It's really a pleasure to present in this forum and discuss some of our experience with optical genome mapping. Although today I am really presenting in the context of a single but very complex case, I'm hoping to convince you of my long-standing belief that every technology in the laboratory is unique and allows for different discoveries, and that one does not replace the other. Between complex clinical presentations and the complexity of the human genome and the alterations thereof, we're finding more and more examples of the value of multiple methodologies in our laboratory. Also, this will be a prime example for the need to keep looking at the genome from a structural perspective, rather than immediately focusing on sequence variants. And when I say that, I plea for the continuing education in cytogenomics and cytogenetics. This is a case that have surely taught us how complex chromosome-level abnormalities can be. So I'm going to talk today about a case of multiple structural variants in a case of atypical Phelan-McDermid syndrome. So we're going to start with the clinical history. This child is a female. She was born at term and came to attention in the neonatal period due to apnea and desaturation events, including hypotonia. The desaturation events actually required intubation, eventually scheduled for a tracheostomy, and was ventilation dependent. She was also G-tube dependent with a significant hypotonia. Upon performing an EEG, there was no measurable seizure activities, and the MRI showed microhemorrhages in the left cerebellar hemisphere. A sleep study showed severe obstructive sleep apnea, and the family history was only, interesting in terms of polycystic kidney disease coming from the paternal side of the family, and an ultrasound for the kidneys of this baby indeed revealed kidney cysts. So because of this presentation, we actually reached out to our research team to do a research-based whole genome sequencing. At that time, our whole genome sequencing was not clinical. It actually just went live at the beginning of January, but at that time, it was still research-based. And in parallel, we actually also sent whole genome sequencing to an outside reference laboratory for clinical testing. So in our hand, the rapid whole genome results on a research basis identified two significant events. First, as you see here, this is the copy number user interface from our genome sequencing, is a deletion of chromosome 4 that encompasses PKD2, likely explaining the familial polycystic kidney disease on the paternal side of the family. So we know now, and certainly upon finding out the renal ultrasound abnormalities with the cyst, we can kind of explain that our patient has inherited the paternally derived copy number deletion of the PKD2 locus on chromosome 4. In addition, we saw a very big terminal deletion of chromosome 22. It encompasses SHANK3, which is the critical region for Phelan-McDermid syndrome. So at face value, this patient was diagnosed with Phelan-McDermid syndrome. So let's talk a little bit about Phelan-McDermid syndrome and the clinical feature thereof. We definitely observe neonatal hypotonia. There's usually absent to severely delayed speech, developmental delay, dysmorphic features that I've listed right here, a moderate to profound intellectual disability, and a decreased perspiration that results in a tendency to overheat. In terms of diagnosis and testing for this particular syndrome, it is usually manifesting at a heterozygous deletion of chromosome 22q13.3 with involvement of at least part of SHANK3. We can also see in this particular syndrome pathogenic variants in SHANK3. And in general, the recommendation is really to order a karyotype, a G-banded analysis, to rule out the presence of a ring chromosome 22. So this is the proband chromosomes when the clinician did order cytogenetics G-banded analysis. And you can see right here immediately that there is, you can see a deletion on chromosome, on the long arm of chromosome 22. This is actually a kind of a panel on chromosome 22 where you can certainly appreciate the deletion on the abnormal copy. In addition, however, you can see that one copy of chromosome 3 and one copy of chromosome 12 actually are involved in a translocation, which at this resolution looks balanced. So we have a translocation between the long arm of chromosome 3 at bed Q27 and the long arm of chromosome 12 at bed Q23 in a balanced translocation. So this was really interesting because well, a lot of interesting question. We know that the phenotype of this patient, in terms of the neonatal period and the significant, desaturation events and the ventilation needs, we were wondering whether this particular balanced translocation was in any way related to some of the phenotype, and kind of teasing out what is consistent with the diagnosis of Phelan-McDermid syndrome and what is not. So because of this, we wondered if the breakpoints of this balanced translocation could be in phenotypically relevant genes. And to address this and to see what else is going on in the genome, we decided to do optical genome mapping, which certainly has better resolution than traditional chromosome analysis. We also wanted to include parents in this testing so that we can see if this balanced event was de novo or inherited. So when we thought about this, I mean, this is a patient that has had whole genome sequencing and chromosome, a G-banded analysis. So we were looking in terms of suggesting to do optical genome mapping, which at the Nationwide Children is in the research basis right now. We were looking for what is the needed technology. Well, we wanted to identify clinically significant structural variants and copy number variants because we wanted to investigate both the 312 translocation as well as the deleted 22. We wanted something that's high resolution, automated, and labor efficient. And we are, very fortunate to be able to do optical genome mapping. So I know for this audience, it's probably repetitive to really talk what is optical genome mapping, but just to give an idea, this is an imaging-based technology to perform an unbiased assessment of genome-wide structural variants. It involves site-specific fluorescent labeling to generate large maps spanning each genome. These maps, they're kind of an optical banding patterns, and they are kind of compared over long contiguous reads. So you isolate your high molecular weight DNA, you label it with fluorescent tags that kind of presents as an optical map or an optical banding pattern. You linearize the DNA and you kind of scan the DNA for these optical patterns, and you generate what is called digitized molecular barcodes. Then you bioinformatically kind of align to a reference genome, and discrepancies in the labeling patterns are identified. So just a quick reminder of what this technology can reveal. For example, it really can reveal anything that is structurally aberrant in the genome. You can see your insertions because you're labeling, as I said, you're labeling a map is going to be different. You can see deletions. You can see translocations where you start with a chromosome and then you pick up the label of another chromosome. You can see inversions because it gives you a different banding map. You can see tandem duplications on the chromosomes, and you also can see repeat expansion, specifically as it relates to trinucleotide repeat disorders. So to go back to the proband chromosome, just a reminder, upon chromosome analysis, we saw a deletion of chromosome 22 and apparently balanced translocation between chromosome 3 and chromosome 12. So these are the optical genome mapping results from the Bionano, and I do want to give a huge thank you to Katie Miller and Anthony Miller, who are the right now the owners of the optical genome mapping in our technology development group at the Institute for Genomic Medicine. So let's start with the proband. The proband, we can definitely identify the 312 translocation. If you see here with my arrow, we can see the deletion on chromosome 4 encompassing PKD2. You can also see that there is a deletion on chromosome 22, but that deletion is also, there is an involvement of a rearrangement between chromosome 22 and chromosome 17, which was not apparent on neither whole genome sequencing or on cytogenetic analysis, G-banded analysis. You can also see for the father that the father is a carrier of the 312 translocation. So this translocation is not de novo. It is, also with the deletion of chromosome 4, they are now both paternally inherited, and the mom did not show any clinically significant copy number variants. So let's kind of zoom in on the finding from optical genome mapping for this particular patient. So we can certainly see the deletion of the PKD2 locus, explaining her renal cysts. We can also see the 312 translocation. So this is kind of the proband in blue. You have the reference sequence for chromosome 12, you have the reference sequence for chromosome 3, and you can see that the proband kind of starts with 12 and then goes up to 3. So therefore, confirming the translocation 312. But more importantly, we also seem to have a more complex than previously thought, rearrangement between a chromosome 17 and chromosome 22. So what we think happened from the OGM data is that there is a portion of chromosome 17 that tandemly duplicated and then got inserted into chromosome 22. We think from the closeness of the breakpoint between the deletion and the actual gain of chromosome 17 interfacing with chromosome 22, that gain of chromosome 17 is actually has been inserted onto the deletion, the deleted chromosome 22 homolog. It can be actually confirmed with OGM by phasing, but you can see how close the structural collar with the deletion collar is. And so we think that these two events are pretty linked. And this gain of chromosome 17 is actually about 800 kb, so it would not be visible by chromosome analysis, and therefore we did not really know that chromosome 17 was involved at all. So the summary of findings for this baby, there is a deletion on 4q encompassing the PKD, the polycystic kidney disease 2 locus, and that is paternal. There is a balanced translocation 312, and we know that it's balanced from the OGM data, and it's also paternal. We also know that there's a rearrangement between chromosomes 22 and 17. It is de novo. There is a deletion on chromosome 22 at Q13.2, which accounts for a diagnosis of Phelan-McDermid syndrome. We do have a gain of material from chromosome 17q to 5.3, and we know that that particular gain has been inserted into the deleted copy of chromosome 22. So could there be genes disrupted at these breakpoints that could allow for a different phenotype for this patient? In order to kind of, confirm the finding between, OGM and whole genome sequencing, we are very lucky in that NCH, in our technology development group, we have also the long read sequencing capabilities. And so we ran a PacBio long read sequencing on this patient. Interestingly, we can see the chromosome 3 and 12, both by OGM and also by the PacBio long read sequencing, and we can identify the affected genes. and they're both the same, so that these confirmed each other. And we did not find that the disruption of these genes could cause any of the phenotypes that we're seeing in this patient. These are the kind of the outlook, the user interface for PacBio long read sequencing, and you can definitely see that we can see the deletion on chromosome 22. We can see the deletion on chromosome 4. But interestingly, the PacBio, although if you go and look at the reads, you can find the chromosome 17 and 22 rearrangement, but the structural variant caller did not pick it up. So only OGM really was able to see it. So I wanted to share a tale of several technologies just to let you know what are the alterations that are present in this patient and what are the methodologies that were able to give us all the information for this patient. I just wanted to give a quick information that from a single nucleotide variants or indels, that patient did not have any pathogenic or likely pathogenic or even VUS variants that are sequence related. So this patient really is a kind of a structural genome, abnormality case. So the alteration types that are present, there is a loss on chromosome 4q. There is a loss and a part of an unbalanced translocation on chromosome 22. There's a gain, an unbalanced translocation on chromosome 17, and there's a balanced translocation between 3 and 12. When we did whole genome sequencing, we can see both losses. We did not detect neither the chromosome 17 involvement, nor did we detect the 312. The reason why I say we can confirm is because that happened in hindsight. So if you go back to the reads and you know what you're looking for, then yes, you can see the reads spanning those rearrangements, but none of the structural callers or the CNV callers actually called it. By G-band analysis, due to of course the resolution, only the deleted 22 was seen, and also the balanced translocation between 3 and 12. If we go to the long read sequencing, chromosome 4 loss was seen, chromosome 22 loss was also seen. We could not detect by the structural variant caller any of the involvement by chromosome 17. We can also, in hindsight, go back and confirm it, but that didn't count. But we did see the chromosome 3 and 12 rearrangement. So of course, hindsight is 2020. After we, like I said, once we kind of identified what was going on using optical genome mapping, we can go back and kind of more carefully look at a read and say, okay, yes, in hindsight, we can see it. From the whole genome sequencing, this is a chromosome 17. You can see here a duplicated blip right here. And this is the copy that is actually gained. It doesn't encompass any genes of interest. So this neither was it called, nor upon visual inspection of the copy number from whole genome sequencing, nor would we have paused on it. There's also a deletion you can see here, but also it did not have any genes of interest so we also did not pause on it. These are just to show you visually the read. The reads do support from the PacBio. They do support these rearrangements. But however, it really wasn't the 2217 was not called neither by whole genome sequencing nor by long read structural variant caller. So I just wanted to summarize that this proband has multiple structural variants identified by rapid genome sequencing, optical genome mapping, PacBio long read sequencing, and traditional karyotypes. As I showed in the table, we actually did show that optical genome mapping was the most comprehensive technology to see all the structural abnormalities that are present or potentially present in this baby. While her presentation seems to be more severe than typically seen, there are actually atypical cases similar to hers that have been observed. And this was via our clinician, our geneticist, personal communication with Dr. Phelan, Katie Phelan. The familial PKD was explained by these studies. The family was counseled regarding risk of unbalanced offsprings or pregnancy loss resulting from the 312 translocation, and surely the family has been referred to appropriate specialties and patient advocacy group in terms of her Phelan-McDermid clinical diagnosis. I do wanna say, at the end, as I close out here, is that the things to consider for this case is, although at Nationwide Children we have a lot of technologies and we can kind of try this and this, we need to consider global equity, which is really doing the right test for the right clinical situation. My biggest plea here is to continue to educate on the importance of understanding the science of cytogenetics and genomics and the large-scale genomic aberration, and try to kind of, evolve as the technology of cytogenetic science evolves, and we know that optical mapping is kind of the best candidate for the evolvement of this for the evolution of this technology. I really do emphasize the importance of educating on cytogenetics and continuing to keep that expertise. This is, the effort of a big team. I do want to acknowledge Erica Mackey, who is our LGG fellow, who's writing up the case. Marianne Matthew, who's a colleague clinical director at IGM, the cytogenetics team, the variant interpretation team, the whole NGS team, and the other clinical directors and the genetic counselors. This is in collaboration with Dr. Bimal Shadari, who has a whole genome research project, and Lauren Walker from the clinical team. And also very, very huge thank you to the technology development who are doing, in collaboration with a clinical laboratory, a proof of principle for the use of optical genome mapping across many diseases. One of them is we are involved in epilepsy. The other is for neonatal whole genome project, as well as heme malignancies. And with that, I will stop and we'll look forward to a discussion later on. Thank you.